So now we are moving on into, still in the golden age of microbiology, but we're going to move toward Robert Koch. Robert Koch was probably fairly close to being as significant of a contributor as Louis Pasteur. He was German, a very famous man that focused on etiology. So he was the father of etiology. And etiology means disease causation. So what was the cause of the disease? He was very interested in that. He and Pasteur kind of went hand in hand constantly on things that they did. For example, Robert Koch may have been studying the same thing as Louis Pasteur. And Louis Pasteur wanted to discover that one microbe caused one disease. Well, Robert Koch was able to solve that problem. Robert Koch may have been studying very deeply into rabies. Well, Louis Pasteur is the one that wound up with the cure for rabies. So they went back and forth quite a bit. They were they were around in the same time frame. So it was kind of a rivalry there. They were very adamant about their job and were very good at it. So let's go ahead and talk about Robert Koch a little bit and some of his major contributions. So he was able to go further with Pasteur's discovery of the germ theory of disease. And he was also able to discover that specific diseases were caused by specific germs. And any sort of disease-causing agent we now know as a pathogen. Thanks to most of these individuals that worked in this era. So a pathogen is a disease-causing agent, and he studied a number of significant pathogens, one of which was anthrax, that he is the most well known for. So the disease-causing agent, the bacterium that causes anthrax, is known as Bacillus anthracis, and he studied it quite intensely in his time frame. That was one of his very first things that he discovered and gave us what we know as, let me get my eraser off here, Koch's postulates. Now with Koch's postulates, he was basically able to prove that one disease-causing agent causes one disease. Those postulates are on page 14 as it shows. Let me toggle over so I can make sure you can see it. Yes, it's right here. And the first one is the suspected causative agent must be found in every case of the disease and be absent from healthy hosts. Number 2, the agent must be isolated and grown outside the host. Number 3, when the agent is introduced to a healthy, susceptible host, the host should get the disease. Number 4, the same agent must be found in the diseased experimental host. This basically means that I can take, if it walks like a duck, talks like a duck, it has to be a duck. So, whenever I draw blood on my patient, I should find evidence of this specific disease-causing agent. That's the first one. If I draw blood on a healthy individual, it should not show signs of that disease-causing agent. Second one, must be isolated and grown outside the host. I should be able to take that blood I drew and be able to culture or grow the bacterium, for example, it may not be bacteria, that is causing the disease. I should be able to take that culture and infect a healthy individual. And they should get the same signs and symptoms of the same exact disease. I should be able to take the person that I should be able to take the patient that I've infected, draw blood, be able to find the exact same thing that I saw originally. So if that makes sense, those are your 4 Koch's postulates. They're very important. And this proves that one disease-causing agent causes one specific disease. This is something that evaded Pasteur, and it drove him nuts. Let's talk about how he discovered this, because there is a person that is very significant to that discovery, and she's shown here. Her name is Fanny Hesse. Now, here's an interesting thing. At this point in time, they did not have any typical way to grow bacterium. They grew it in broth. Remember, we keep seeing this over and over. We grew bacterium in broth. So he was ahead of his game, and he came up with what we call the potato slice method. Worked pretty good. You slice a potato, grow your bacteria on that slice. However, it's mainly starch, and not all bacterium are going to grow well on starch. There's actually quite a few that are not. That's very selective for bacterium. They'll just enjoy eating starch, so to speak. So therefore, this was very limited and caused a problem with growing a lot of bacterium. So they were still trying to discover what would work. So they used gelatin with their broth. Well, they couldn't get it to solidify, because here's the neat, crazy thing about gelatin. As soon as enzymes start to digest it, it turns to liquid. So it wouldn't work. So one of his apprentices, and we all know we take our work home sometimes, was talking to his wife, and he was very frustrated. He said, we can't get this to work. I don't know what we can use. I'm very frustrated. So she came up with a brilliant idea. Why don't you just use the material that I use? It's called pectin. And pectin is what she would use to can her jams and jellies. It sets up, it's congealed, and it stays that way for a very long period of time. Nothing changes it. So she thought, hey, this is going to be a great idea. I think you can use it. So he takes it and he uses it, and it works great. And it comes up and it creates what we know today as what we call agar. And this is what we're going to use today to grow our specimens and culture them in the lab. The lady's name was Fanny Hesse. She was just a housewife. However, she is smart enough to have figured out what would work for us to grow bacterium and other types of organisms and be viable. So that is how he was able to go about growing his bacterium and isolating it, because you can't isolate it in a broth. You can isolate it, however, whenever you have agar. And we're going to talk a little bit more about isolating organisms and things of that nature as we go throughout the semester. This is just basically your introduction. All right, so let's talk a little bit more about some of the amazing things that Robert Koch did. He's got a lot of firsts that he did. Remember, he's the father of etiology, disease causation. And you'll notice here all the many things that he has done. Simple staining techniques for bacteria cells and flagella. He was the first one to do that. First photomicrograph of a bacteria. First photograph of bacteria in diseased tissue. Techniques for estimating the number of bacterium in a solution. He was the first one to do that. Use of steam to sterilize growth media. First person to do that. Use of petri dishes to hold solid growth media. First one. Laboratory techniques such as transferring bacteria between media. This is going to be part of our aseptic technique you're going to learn soon. Elucidation of bacteria as distinct species. Basically, he learned elucidation, kind of euphoric event, was able to discover bacteria as a distinct species. Remember before, we just understood them to be germs or microbes, and we didn't really understand there was one specific bacterium for one specific disease. So there's a lot of discoveries going along, or happening, I should say, in this time frame. Okay, so we've gone past Robert Koch. Now let's go ahead and take a look at some other significant individuals that are going to be known in this time frame. On page 15, we start with gram stain. Now, you'll notice here, this is the gram stain. It's a very famous type of staining procedure that's called a differential stain. And it's called a differential stain because it's going to ID two main types of bacteria. And this is bacteria. We still use the gram stain today to be able to identify and break our bacteria into two main types based upon their cell wall composition: gram positive and gram negative. You see in the picture, you'll see purple and pink. Once they've gone through the procedure, gram positive stains purple, gram negative stains pink. And I'm not going to go through all those details of the procedure just yet. We will do a gram stain later on in the semester, and we'll talk about it some more. But that is a gram stain. The person that come up with it is Hans Christian Gram. And he discovered the gram stain. Major contributor to how we organize and classify bacteria today. So at this point in time, we've learned a little bit of disease causation, things of that nature, and suddenly we are going into some very important aspects such as preventing infection and disease. There's a number of people that are going to start the sanitation movement. So I consider this classified as the sanitation movement. So let's start off with an understanding that we have what's called HAI. Healthcare associated infections. Another term for it is also nosocomial that you may hear. I don't know why or who came up with the nosocomial term, but it's health acquired infections. And we understood in the 1800s, because we're kind of rolling along in the 1800s now, 1800 to 1850, early turn of the century of 1900, this is whenever we started coming about with most of the things that we accept today as far as healthcare goes, and starting to begin some of our treks toward a healthier style of living. So in the 1800s to 1900s, we still understood that you went to the hospital, you're probably going to die, not necessarily from what you had, but possibly from an infection that you acquire at the hospital. And we still know today that sometimes some of the worst infections you can get are hospital acquired infections. So they're still very real. However, they're quite different from what they may have been 150 to 200 years ago. So we had some very important people that came along and created our sanitation movement, one of which was Ignaz Semmelweis. Let me make sure I spell his name right. he was a very important man. He worked in Vienna, and he was a physician and a teacher because he worked in a teaching hospital in Vienna. Now, at this point in time, we had very little understanding of disease causation and things of that nature. But also remember, at this point in time, we had no running water. We had no sanitation, such as toiletries, things of that nature. We didn't have toilets in buildings. We didn't have running water in buildings, not even in hospitals. This did not exist. What you see when you look at old castles and things of that nature, think about that, because that is what you had. Okay? Water was typically community water. It was drawn out of a well. There was no plumbing. That was not existent. Bedding, things of that nature, was privileged. We've got to remember that the way of living in the 1850s was not the same as it was today, or is today. So we have to remember those things. So, Ignaz Semmelweis was in a teaching hospital, and he was responsible for a number of doctors that were that were studying under him to become doctors, I should say, students. And they would go through and perform midwife practices, childbirth. And what would happen is a lot of them, the women would catch what we call puerperal fever. Let me see if I can find that word right quick because it's really hard to spell. There it is. P-U-E-R. Puerperal fever, and it was due to a bacteria that was in the family of, or I should say genus, of Streptococcus. So it was a streptococcal infection that caused puerperal fever, and they would become very sick, very ill, and die. So the women would come in for childbirth, and we also realized in that time frame that if they came in for childbirth, then they would die from this infection, and that was very common. So a lot of women that had childbirth in those days would just have birth on the farm and things of that nature. It was safer. But the other half of the hospital had midwives, and he thought it was very interesting that the midwives did not have near as bad of an infection rate and a death rate as he did. So he tried to figure this out. He started studying this intensely, and he learned a few things about how the midwives performed their childbirth that was different from what he did. And he really studied it quite intently. Now remember, in this point in time, even your most intelligent people, they thought that there were spirits that were guarding people, and things could possibly, you just have spirits that cause the infection. So one of the things that the midwives did was whenever a child was born, they would ring a bell. Semmelweis at first thought, okay, well, maybe it's the bell ringing that is warding off the spirits. So they tried that. Of course, we know that's not going to change anything. So he didn't have any effect. That had no effect, him ringing the bell. So what he did, then he noticed that his students were working with cadavers in the morning, and then they were performing childbirth in the afternoons. Well, do we wash our hands in the 1850s? No. We might bathe once a week, and that'd probably be the very privileged people. So it would be very, very common for these individuals to probably still have cadaver particles is what he called it, or bacteria on their hands. He called it cadaver particles. And he said if he could remove the cadaver particles, he thought that the childbirth risk associated of catching puerperal fever would go down. So he took chlorinated lime water and made his students rinse their hands before they went in and worked with childbirth in the labor division. The death rate declined and people were living and changed the numbers drastically. Now, this would probably be something you would think the Semmelweis would be praised for, and this would be a significant finding. Well, unfortunately, he was very adamant about everyone needing to wash their hands. He became quite adamant and quite prolific. I don't know what to say, but he was a bit of a extremist on this. So therefore, he was laughed out of the whole department by his directors and his peers within 4 years of this happening. And then shortly thereafter, he was committed to an insane asylum, and that is where he died of a streptococcal infection. So it's quite ironic that Semmelweis died in an asylum with the same infection that he treated and was able to prevent. I think that's rather ironic and kind of sad in a way, but that was Semmelweis. He was one of our main instrumental characters in the beginning of leading to our sanitary movement of washing hands and keeping ourselves clean and sanitized. Now, the next person I want to talk about with our sanitation movement would be Lister. Joseph Lister was also an English physician, and he was also a teacher, and he taught doctors. His problem was he was very concerned about sepsis. What is sepsis? It is basically infection. So he wanted to create something for antisepsis against infection. Now, what his problem was is that he was doing lots of amputations. We also know in these days, whenever you had a limb amputated or anything amputated, you pretty much counted on the fact that you were probably going to die from the infection. Why? Because we did not have any, zero, none, antibiotics until the first antibiotic, which was penicillin, which came out around World War II. So whenever you start thinking about it in that aspect, that was not that very long ago. At this point in time, we had no antibiotics. We had very little understanding of disease causation and things of that nature. These people are our founding fathers starting this and starting the clean sweep of a sanitation movement. So we have Lister that comes about in his antiseptic technique. So he decides that he's going to come up with something that he can treat his patients with that are getting amputations in order to reduce the risk of amputees. So he comes out with a product that is carbolic acid. Okay, this is all on page 16, just so you know, carbolic acid. So his carbolic acid spray is basically what we know now as phenol, which is our first antiseptic, and we base all of our antiseptics off of. He sprayed this on the wound, and he kept spraying it on it as it was healing, and he noticed a great reduction in infection of wounds. This was a major breakthrough. Now, later on in life, we have learned that carbolic acid is a little bit carcinogenic, which meaning it can cause cancer, and it's dangerous, so we don't use it anymore. However, it was a major contributor to our first beginnings, and it's amazing to see what that has done for a lot of individuals that otherwise would have died having amputations, or having an amputation done. All right, so now we've talked about Lister. We're going to go on and go to our next individuals. So a very, very important person in the sanitation movement is a wonderful woman by the name of Florence Nightingale. Most of you will know Florence Nightingale, or the name, because she is so incredibly well known in the field of nursing. She was a very dedicated English nurse that introduced cleanliness and antiseptic techniques into the nursing practice. Now remember, at this point in time, we're talking 1820 to 1910, she was very ahead of her game. Nurses were not revered in the same manner that they are today. People that were nurses really weren't taught much. They just went out and here's your materials, go be a nurse. Also at that point in time, there were a lot of nurses that died and didn't last very long, so that it wasn't something that was a really prestigious job. A lot of times, a lot of wars were fought in this era, and if you were a nurse and you were recruited, you basically went to die. It was not a very nostalgic time for medicine and for nursing especially. Florence Nightingale was actually a very aristocratic family, very well known, very popular, and her parents hated the fact that she was going to become a nurse. They did not want her to be a nurse. They wanted her to marry a very distinguished gentleman and have a distinguished lifestyle. They did not want her to have a very posh lifestyle. They did not want her to have the lifestyle that she chose. She was already fairly well known in her nursing career when the Crimean War began. And she was recruited for the Crimean War. In 1854 to 1856. She sets foot on the front lines and immediately says, well, there's no wonder these soldiers are dying, because their sanitation and their health is inadequate. So the very first thing she said is she needs to get these soldiers into a clean area. They need fresh beds that are not covered in filth and dirt. They need fresh clothing, again, that's not covered in filth and dirt. They need to clean the location and remove the sources of infection. She said another reason why they're dying is because they're thin, they're underfed, they're malnutritioned. So she was instrumental in in getting better food and getting better conditions for soldiers. She was known as the Lady of the Lamp, was the saying that they would call her, because she would go around at night watching the soldiers and taking care of them in the Crimean War. She was diligent and never stopping on what she did. It was amazing how she reformed hospitals and reformed the way we look at sanitation in the medical field today. She was very, very significant into those sorts of things. Created the very first school for nurses. the Nightingale School for Nurses. That was the first one in the world. I was trying to think of some other interesting things about her. I know she was found ill. She was sick for a good portion of her life expectancy. I shouldn't say life expectancy, for a good portion of her life with an infection that she had gained during, I want to say during the Crimean War, but I can't remember. It was at some point in her life, but she never stopped continuing on and getting improvement of sanitation and health for hospitals. It was just an amazing person just to get to be able to read about her and learn more about her. She was so diligent and never stopped, never ceasing in what she really believed in. Okay, so we've gone through Miss Nightingale. Now let's go ahead and go to our next person, which is John Snow. He was another amazing person that is mainly known for our sanitation movement in general, because he was hired for the original Soho district. Sometimes you guys think Soho district in the U.S. There's a Soho district in London, okay? He was hired to discover what was causing disease in the Soho district in London. And what he discovered is that cholera is causing the infection. And the cholera is coming from the water. He did a number of different studies, and he said the cholera, I think, is coming from the water in that community. So he, again, remember, we don't have running water. We don't have pipes, plumbing. That's not something that's common. So we have a community watering hole. And he said, remove the spout and remove the source of the ability to get water from that area. He said, it's coming from this specific area. I need you to remove it. And they did it. And when they did, they started seeing the cholera cases go down and people ceasing to be sick. So what they decided to do, thanks to John Snow, is plumbing, toilets, things of this nature. Sweeping the streets did not exist before this time. So they had dead matter in the street, hay in the street, trash in the street. All of this was not something people did. We did not clean up our trash. Okay, I know it sounds really disgusting, but we did not clean up our trash. This is what started that movement. Okay, our lifestyle and way of living is called the European lifestyle for a reason. And it's called that because of this person and his beginning of that actual sanitation sweep right there, from beginning with taking that water and removing it from that community so that they could actually realize that they needed to have cleaner, safer water to be able to drink. So that study, he was able to study the cause of the disease, how it traveled through a community, and the cause of the infection. So therefore, he is basically our father of epidemiology. Sorry guys. Alright, so epidemiology is the study of the occurrence, distribution, and spread of disease in humans. Shouldn't say in humans necessarily, but it is the study of the occurrence, distribution, and spread of disease. He was very instrumental in infection control. Okay, so let's go to our next person that we want to talk about. So while everyone else at this point in time, so far we've talked about, it's mainly a focus on bacteria, we also have someone studying other things. Viruses. So we'll start with our first guy named Edward Jenner. And he came up with the very first vaccine. So we're talking around 1800, 1796. Around that time frame, we have this English physician that it lives in a small community, and he discovers that smallpox is the sad thing about smallpox is it killed 1 in 3 children by the age of 3 in this time frame. You start thinking about that, that's very, very scary. 1 of 3 by the age of 3. So you can notice that smallpox is very prevalent in the society, kills a lot of people. You got to realize that at this time frame, we still have a lot of death and mortality due to infection. And we don't really have that anywhere near that drastic anymore at this point in time. That's why we're so concerned with COVID. We don't have anything so drastic and out of our grasp of our knowledge until just recently. So he went through and was first one to discover, hey, I think I can, wait, let me back up. All right, so he's here watching smallpox, noticing that it's killing so many people and it's horrible. But he also observes the milkmaids. And he thinks, this is interesting. They come up with these splotchy rashes that are similar to smallpox on their skin. However, they're fine. They don't get sick with smallpox, and they never really have any major problems. So he observes this and takes a scab from a person that has received cowpox, which is very similar to smallpox. And what's the milkmaid? This is what the milkmaids have been getting as far as what's on their skin. So he takes that scab, grinds it down, runs a needle and thread through it, and then I'm going to get a little crazier because the CPS would definitely be all over him. Takes a child, I believe it was one of his students, his apprentices, children, and he runs it through the skin and inoculates the child with the cowpox. He gets sick for a few days, but then starts to feel better. So then he's like, okay, this may work. Well, we have to prove it. So how do we prove things like that? We go and stick the kid right in the middle of the smallpox community. So we make sure he doesn't get the infection. Yeah, CPS would probably be all over him by now. So he does. The child does not get smallpox. So therefore, everybody's running to Jenner to get the cure so that they can be treated for smallpox. And this is our first vaccine. It originates from a Latin term, vaca. Now, if you know Spanish, you know that means cow. So vaccine came from vaca, meaning cow. Why? Because the very first vaccine was cowpox that was used to treat someone for the smallpox disease. And that's very common to use something similar, a modified live vaccine, that's what that's called, to treat for a virus. All right, so we've gone past him and we're going to go, actually Jenner began the field of immunology. Let me make sure to mention that. He was our father of immunology, the study of body's defenses against pathogens. All right, the next one we want to look at is Paul Ehrlich. Make sure I spelled his name right. Yeah. He is our father of chemotherapy. He created what he called the magic bullet. We call it the magic bullet because it was able to destroy pathogens while remaining non-toxic to humans. And we didn't think that could be possible. And he created his specific one that he called the magic bullet was called salvarsan. It was an arsenic-based drug, okay? And he had said that it would cure syphilis, which is a very, very nasty STD. Just an interesting tidbit, Al Capone died from syphilis. They actually released him from, I believe it was Alcatraz that he was in, but wherever he was at in there, Ellis Island, because he had stage 4 syphilis and was deemed as someone that was no longer a threat to society. So, he died from syphilis. Very sad thing. But syphilis is very dangerous and very, very scary. But he had, Paul Ehrlich had discovered a disease called, a chemotherapeutic agent that was called salvarsan that was supposed to cure syphilis, which we know now doesn't work in that manner, especially because it's arsenic-based compounds, and anything with arsenic is also very toxic to humans. So he is still called the father of chemotherapy. However, his magic bullet wasn't quite as great as we would hope. Alright, so we've gone through Ehrlich. I'm not going to go through the modern age of microbiology here. Let me go ahead and talk to you guys. I know there's a few other people I want to talk about. Let's go ahead and mention Ivanowski. Where's Bajaric? There he is, in Bajaric. Okay, I'm going to go ahead and finish everything up because I only have a little bit left to talk about. So let's go ahead and let me pull my whiteboard up here. We have Ivanowski. And let me spell his wonderful name correct. Bajaric. Yeah. These two discovered the first virus. And while we have Louis Pasteur discovering our, no, I just went blank. We have Louis Pasteur over here studying and discovering things, and these other people over here discovering things. It's very interesting. Oh, yes. We have Louis Pasteur over here studying wine. One of the other main crops of this time frame is tobacco. So if you want to really upset your major owners of this time frame, tear up their tobacco or their wine. Of course, wine in those days, ugh. No, I don't even know if I want to drink old wine like that because I think of how it was processed. Nobody wore shoes and how they processed it. They stomped on it with their feet. But anyway, tobacco mosaic virus is a very predominant thing that we need to look at because it's the very first virus that was discovered. And what they did is they used a filtrate. And let me see if I can find it in here. I don't know if it's around here. And they filtered the virus out using a liquid. I mean, that's how you do a filter. Just think about a coffee filter that you have in your house. It's going to be basically the same thing. And by doing so, they were able to filter out the virus and identify it. Well, at that point in time, they weren't sure if it was liquid or what may be going on. They had no idea about anything concerning viruses. They just discovered the first one. But they did it through a filtrate system. Therefore, it makes sense that they thought it might be possibly a liquid. So when they named it virus. Well, virus translated means poison. And a lot of times when you think of a poison, you think of a liquid. And they didn't have a good understanding of viruses and things of that nature, so they thought it was a poisoning the tobacco plant. And so it's very interesting to look at that and that term. I'm trying to think of anybody else that I need to look at. Alexander Fleming, he is our father of pharmaceutical microbiology. He discovered our very first antibiotic known as penicillin. We'll talk about him later on in the semester. Another person that I want to mention, as all of this is going on, is Walter Reed. And he's not anywhere in your textbook. That's why I say you need to make sure to watch your lectures. Okay, so let's talk about Walter Reed. He's a Navy man. He was one of the major people that were in the military that was a microbiologist. Now, let's think back to this time frame. In the late 1800s, early 1900s, we had 10 years only, but one major historical event that happened in the United States were the Great Cattle Drives. Now, the Great Cattle Drives were very heartbreaking, very tedious, very hard, but one thing that they helped us discover in a microbiology sense is the term vector. Now, what in the world is a vector? Something that can carry disease. They don't get infected, but they can carry it. Okay? If you're a vector, that means you're not necessarily showing signs and symptoms, but you're carrying the infection. You can give it to someone else. It helped us discover that there are vectors in this world that are known as intermediate hosts. An intermediate host may be a mosquito, a tick, a flea, things of this nature. So that helps you possibly to understand what may be an intermediate host. So therefore they're carrying the disease, but they are not going to show signs or symptoms or anything. All they do is transfer it from one species to the next. Now, I'm talking about all of this stuff to get to an important part of history. So, where at the Great Cattle Drives, we've discovered that a vector can carry disease and things of that nature. Along the same time frame, we have the building of the Panama Canal trying to occur. A number of other countries, such as the French and the English, tried to do it. When they would go down to the Panama Canal, their construction science team would get incredibly sick and die from a disease known as yellow fever. They didn't have a good understanding of it, so they thought the area was cursed and to leave it alone. It was best that we not try to build the Panama Canal. We just go ahead and go around South America like we've always been doing. Because we don't need to push the, if you want to call it the vital force. Okay, the spirits that are down there, it's cursed. It's bad, so we need to leave it alone. Well, America was a very young country still at this point in time, and you know how they are. They're going to prove everybody wrong. So they sent their team of microbiologists down to study the disease and the cause of it. Walter Reed was one of the primary individuals, and Walter Reed discovered yellow fever and discovered that there is a mosquito actually that is carrying this disease. And he said if we put nets and protective coverings over our crew, our boats, and things of that nature, we can build the Panama Canal, and we will not become infected with this virus. He was correct. So therefore, the Americans were able to build the Panama Canal when other countries failed. To me, that's a major significant thing to look at, and I think it's very interesting to see how science, and specifically microbiology, has affected history in such a major sense. Now, this is all for Chapter 1. And we will cover Chapter 3 next. Well, I skip Chapter 4 and come back to it, and I never cover Chapter 2. So we're going to go to Chapter 4, and then we'll come back to Chapter 3 for this unit. So this is all we have for Chapter 1. I hope you've enjoyed studying some of these historical contributors of science and microbiology. Don't forget your paper that you'll need to write, and also to take your quiz over the Chapter 1.